very briefly, I want to talk about my positionality as a non-Indigenous researcher. So I am non-Indigenous working in solidarity with and learning from the North Fork Mono tribe, and this project has developed through ongoing conversation with the chairman and stewards, practitioners. And I'm also an international student from China. So growing up, I was surrounded by my own forms of cultural ecology. So I was talking about my upbringing and learning of my own forms of cultural knowledge in school and also in poetry and in practice. And those backgrounds shaped how I approach ecology, and it reminds me that knowledge does not only exist in schools or scientific institutions. It also lives in communities and languages and long relationship with the land. So I view my role in this project as not to represent tribal knowledge. The goal of this project is to hopefully support tribal monitoring and future grant efforts with data, um, and to respect data sovereignty. All the results will be interpreted collaboratively with the tribe, and all the raw data will be shared with tribal partners prior to any publications. So with that, I'll move into introduction and situate us in a broader historical context of the Sierra Metals. The Sierra Metals has always been shaped by indigenous fire stewardship. They were actively stewarded by indigenous communities through cultural burning for generations. However, following California statehood, a lot of indigenous land deposition happened, and federal fire suppression has disrupted burning practices from indigenous people. And the results from those legislations could be seen as landscape changes, such as metal drying and conifer encroaching into the metals, as well as altered hydrology. And in recent years, started around 2021, we have seen cultural burning slowly being re-recognized, but it still remains structurally very constrained within regulatory liability and institutional constraints. So I think it's very important to recognize that these ecological patterns, such as fire water ecology, is inseparable from governance and policy and power. And one of the places that cultural burning is actively being restored is Benedict Meadow, which is my study site. It is located in Sierra Nevada near North Fork, California. It's on the ancestral homeland of the North Fork Mono tribe, as you can see on the map here. And this map is adopted from the North Fork Mono tribe trail mapping project done by Ron Good, Jess Valdez, and researcher Crystal West. And it's important to note here that Benedict Meadow is the first meadow that North Fork Mono tribe is able to burn last summer on U.S. service land.
Did I jump scare you? Yeah, I bet I did. You make me sick. Weakness.
I don't know how they're getting on, because the waiting room is on. No one got on. I don't know what that was. So sorry. Okay, they stopped. I don't know who it was. It's somebody who's currently on. Please go ahead.
Vivian, can you block them? I think you can kick someone out and block them from coming back in.
No, I did block the original person. No one can get in right now. That's what I'm confused about, because the waiting room is on. And so no one can actually get in unless I let them in. So it's either somebody who's on and I just didn't, they quickly muted themselves. I didn't see anyone active though when that came on.
It's definitely a person.
So, well, all right, so if
I'll
you,
keep-
Letitia and Radhika and Kim, if you can keep an eye out, and let me know, and I can block them. Or I mean, I can report them, take them out, and remove them from the talk. I don't, yeah. So I'm sorry you're wrong, but please go ahead and we'll do our best to keep this from happening again.
It's not your fault. Um, yeah, so I think I just finished the map slide. So going forward, this quote from Chairman Ron was one of the very important motivation for this study. So he said, "A land calls fire, together they call wind, and they bring water." So this reflects a way of understanding ecological processes as an interconnected system. At Benedict Metals, North Fork Mono Tribe has used traditional ecological knowledge to guide their metal restoration. So whenever I talk to Ron and other fire stewardship leaders, they will always say that fire keeps the metal wet and reduces wood encroachment and also promotes culturally important native plants. However, those cultural burning efforts are still very underdocumented in Western science. So the hope for this project is to, while be guided by the tribal knowledge, to combine soil and vegetation monitoring and community-guided interpretations to support tribal-led management and grant efforts with data. And my overarching question is, how does cultural burning revitalizations at Benedict Metal influence and connect soil, hydrology, plants, and people? So I'm curious to understand cultural burning's effects on soil infiltration and soil surface moisture, and how that is connected with the seasonal water table at the meadow and the meadow's function as a sponge. And the soil and water connection to native plants as well as encroaching plants, their regrowth as well as the reducing of encroachment. And lastly, I'm also curious to understand how the indigenous fire practitioners observe these ecological changes and how they feel about the revitalization effort. So going to the research approaches, first I'll talk about soil and hydrology. And here the key question is, how does cultural burning influence soil processes that support meadow water and plants? To understand that, I'll use soil moisture probes to record seasonal dynamics of near surface water and shallow groundwater wells to record changes in water table through the growing season. And lastly, infiltration tests, which is pictured here, to see how quickly soil is absorbing water after burn. And all of these will be repeated measurements to track any seasonal changes that we can see. And to link this to plant, we'll take soil samples from inside and outside of the piles to look at the stable isotope, such as carbon-13 and nitrogen-15, using RIMS, and that will help us understand the plant nutrient use and how soil is connected to plant recovery. And going to plants, the first question that I'm very interested in is, how does cultural burning influence culturally important meadow plants? So there are a lot of culturally used plants near Benedict Meadow, and these are four that I'm hoping to focus on from the North Fork Mono Tribe cultural plant list. So there's bracken fern, which is a basketry material, and juncus, which is also for basketry, blue elderberry for food and the music instruments, as well as gooseberry, a fruit-bearing shrub for food. And I will record under the traditional ecological indicators of plant health, such as how straight are the stem and how long are the shoots, the abundance of fruits, the color of the plants, and phenology, which are also documenting North Fork monotribes TEK. And the second question about plants are how does cultural burning influence the general vegetation structure around the meadow? And to do that, I'm hoping to use walking transects across burned and unburned area to look at percent coverage by species and also document the height and also biomass proxies. For meadow encroachment, which you can see in the photo here, the meadow is this light green area. Here is the burn area, and in between there's some standing tall trees, which are the encroaching cedar and willows. So we'll also look at the recruitment along meadow edges of resprouts of cedar and willow, and also probably using remote sensing to look at the meadow boundary stability. And lastly, similar to soil, I'll take some leaf samples and do stable isotope analysis to see the plant nutrient uptake. And the last component in the interconnectedness of the meadow system that I want to explore are the people. So how do tribal practitioners observe meadow changes? I plan to do semi-structured interviews with North Fork Mono Tribes fire crew members, basket weavers, and meadow stewards who's been working on the land. And the first category of questions I'm hoping to focus on is what kind of ecological indicators have they observed, such as changes in plant diversity abundance and meadow wetness and water flow, as well as wildlife presence and ecosystem health indicators that they think is important. And I'm also hoping to asking about their experience and feeling about the return of cultural burning, and how they feel their relationship with land and their community has been influenced after being involved in their traditional practices. So in summary, this project explores how cultural burning reconnects different parts of the meadow system. So we looked at how fire might influence soil and hydrology, and we'll examine how vegetation responds. And importantly, we'll look at how people observe and interpret these changes. So together, these perspectives will hopefully help us better understand how cultural burning supports both ecological resilience and cultural relationship with the land. So thank you so much. Sorry for the disturbances. And I want to thank the Santa Clara Valley chapter of California Native Plant Society to offer me the scholarship. And I want to thank the North Fork Mono Tribe, Chairman Ron and Christina for always sharing their knowledge and teaching me about cultural burning, and everyone in Fukami Lab who's always supported me. I'm still in the beginning stage of this project, so I'm happy for any feedback and ready for any questions.
Thank you. That was a great talk, really interesting. And again, so sorry about the interruptions.
Really good job navigating through that and just staying on it. So very impressive. I have a very naive question. I was wondering if you no. Are there differences in practice between cultural burning and like fire management that say like the national park would do? I think you're on mute. On mute.
Okay, that's a great question. I'm not very sure how if national parks are doing mostly prescribed burning, or I think I've heard that some national parks are partnering with tribes to also do cultural burn or culturally informed prescribed burn. So I guess I can speak generally about difference between cultural burn and prescribed burning, if that could partially answer your question. I think a lot of time prescribed burning around California focus on fuel reduction and wildfire mitigation. And cultural burning, on the other hand, emphasizing the culture. So, who is burning and for what reason? Mostly the intention is to maybe steward this one plant or steward this one ecosystem. And also, time to time, also about reducing fuel.
Thank you. It looks like you have from Justin Valier, very nice integrated project you're on. And Radhika says, thank you. Great that you are working with the North Fork Indigenous tribe to ask these important questions. And looks like Katie has a question for you, Wrong. Do you want to?
Yeah, hi, you're wrong. This was a great talk. Thank you for providing it. I have a question about the hydrology study. You mentioned that you're going to have the shallow groundwater probes. Does that include things like piezometers and things like that? How do you expect to quantify the data that comes out of that?
Thanks for the question. Yeah, I'm hoping to use piezometers to be installed inside the metal. So the setup of the burns are around the metal edges, and the burn piles are kind of located all around. So for soil moisture, I'll be putting soil moisture sensors in the soil around the piles. But for piezometer, I think it's better to install them directly into the metal along the transect. I am I've never done anything related to water table before. So if you actually have any advice on that, I would happily take them. But so far, I've been working with another grad student who has done a lot of groundwater studies. So they've been really helpful for just updating me on how to set things up and what to expect.
That's great. Thank you.
All right. If there are no further questions, we can thank you very much again. You're wrong. Great job. We can move on next to hear from Kekau Nelson, who is a second-year PhD student in ecology at UC Davis and the recipient of the Don Mayle Conservation Research Scholarship. Her research focuses on prescribed fire in serpentine meadows and how different restoration histories influence native and invasive plant dynamics. By studying both highly managed and minimally managed sites, Kekau aims to better understand how fire can be used as a practical tool for conservation. Her work is driven by a passion for restoration ecology and a commitment to informing real-world land management decisions. Welcome, Kekau.
Okay. Hello, I'm Keikoa Nelson. I'm a second year PhD student in Justin Vallier's lab in the GGE and in Plant Sciences department. And today I'll be talking about my research, prescribed fire in serpentine meadows of varying restoration management histories. So to start off with some background, my research is over in McLaughlin Reserve, which is located in Lower Lake, California. The reserve itself is roughly 7,000 acres in size and has a bunch of different ecosystem types from riparian woodland to blue oak woodland and savanna, grassland, and chaparral. About 1/3 of the reserve itself is covered in serpentine soil, so this has a bunch of habitat patches of endemic and rare plants adapted to the harsh soil there. The site location itself sits on the fault line separating Coast Range ophiolite, which is mostly serpentine soils, from Great Valley Sequence, which is the sedimentary soils. So to continue with background, a bit of a geology lesson in case you don't know what serpentine is. So we have peridotite, which is rock that is rich in heavy metals such as magnesium and iron, but poor in essential nutrients that plants need, such as silica, calcium, potassium, and phosphorus. For this, we have serpentinite, which is the mineral produced during plate convergence and subduction when peridotite is under high pressure and presence of water. So what happens is the plates shift and converge, and then all that magma and that peridotite comes up to the Earth's surface, and we get serpentinite and serpentine soils, so that green rock and that green kind of soil that we see here in this picture. So more broadly, when we're talking about serpentine soils, this includes peridotite, serpentinite, and ultramafic rocks, but California specific, this is large amounts of peridotite, partly serpentinized peridotite, and then serpentinite. So to continue, we have serpentine avoiders, so harsh growing environments impede serpentine avoiders, plants incapable of growing on serpentine soils, resulting in sparse, distinctively smaller vegetation throughout the landscape. In contrast, we have serpentine tolerators, so these are species capable of growing in serpentine soils. California alone has over 200 species of plants that are both endemic to the state and the substrate, with many of these being federally and or state listed, making these meadows important ecological hotspots for conservation. However, serpentine tolerators not only include these really cool native rare species, but invasive ones as well. So rather than being dominated by native forbs and bunch grasses, serpentine meadows are increasingly invaded by annual grasses. So a lot of wild oats, bromes, and more severely, we have Medusa head and barb goatgrass, specifically at McLaughlin. So a lot of these occupy the landscape and are outcompeting our native species and severely altering our community composition. So onto my research, what we're looking to do is we conducted fall prescribed fires in serpentine grasslands of different invasive grass management histories. So we have areas of low management and areas of high management. To break it down, we have our high management. So we have areas where we've done a lot of active, and by we, I mean the reserve managers, have done a lot of active chemical and hand removal of these invasive species. So 7+ years, then we have the same thing, but they've only done this for 1 to 2 years. Then for low management, we have areas where they've done no restoration whatsoever, then areas where they've done some spot treatment and some hand pulling, and then areas with some spot treatment, hand pulling, and then some light grazing from their neighbors' cows. So with this, we created a pseudo randomized block design. So we have 10 matched 10 by 22 meter blocks for a total of 40 plots. And so we have 5 of those laying in the high management 7+ years blocks, and then 5 in the high management 1 to 2 year blocks. So looking at this picture, you can ignore the numbers because that's not where the plots are actually located, and you'll see that on the next slide. But just looking at the management zones, we have in orange all the barb goatgrass control and grazing. We have areas not colored because there has been no restoration management there. We have the areas in purple where it's 7+ years, and then we have the areas in this tealish color where there's control for 1 to 2 years. And then we have the areas in red where there's a historic distribution of barb goatgrass where it exceeded 40% cover. And then these lines show some areas where they've done some prescribed fires of their own. So this is what my research site looks like. We have this yellow line that delineates the management boundary between the two. On the left over here, we have the low management plots. And then on the right over here, we have the other low management plots with their matched high management plots on the inside. And so we have blocks 1 through 10, and then we have all the burn plots labeled in red and all of the control plots labeled in white. So this is what I mean by matched blocks. So we have our low management here and our high management over here. So with that in mind, we have our research question. So what effect does prescribed fire have on species cover and richness in low maintenance and highly maintained serpentine grasslands, both above and below ground? Do low intensity burns reduce the amount of invasive and native seed present in the seed bank? Does prescribed fire promote native growth in these areas? And does prescribed fire alter the soil composition and serpentine ecosystems? So what we're essentially looking at is using prescribed fire as a short-term restoration, focus on invasive grass reduction, and ideally what we'd want to do is couple this with long-term restoration efforts to promote native species restoration. So what we're essentially doing here is trying to create a time frame for land managers so they know when to start doing their restoration. So first we have what our high management plots look like when we went to set up in April. So this is a high management plot. You could see a lot of native forbs in the landscape. And then we have pink flags laid out for where we would set up our plots. And then this is an example of what one of the low management plots look like. So we have a lot of invasive grasses along the landscape, and then we have some native species present as well. So what we did in April is we set up our 10 by 10 meter plots by staking microfiber posts into one corner of the box. So 10 meters was measured out using a measuring tape, and then using a compass, we ensured that all the boxes had equal corners. So we would stand at one of the stakes and then look through the compass to a guiding object to create a 90-degree angle to line up the other corners of the boxes. And so with that, we would put the 2 meter buffer in between the plots. So then we delineate between burned and controlled. So this is what it looks like with all of our posts in one of the high management plots. So we have our one square right here, the 2 meter buffer in between, and then the other square over here. So then this is what the stakes looked like. And so for April 22nd and then April 28th through 30th, we conducted our first round of preliminary observations, and then the second round in June 6th to catch all of those late blooming species. And so what we were doing was recording percent cover for 5 subplots per plot for a total of 200 subplots. And then subplot locations were located 2 by 2 meters away from the posts and 5 by 5 meters away for all of the middle subplots. And then plots were randomly assigned burn treatments. So we labeled the stakes as the following. So the top number is block. The second is management, with LM being low management versus high management being HM. Then the treatment B for burn, and then C for control. And then the subplot number it was. And so April 2025 and in June 2025, what we were doing was putting 1 by 1 meter quadrats on the ground. This is in the low management zone, and so you could see there's a lot of bromus in this plot. There are some native species present. For the most part, it is all invasive or non-native grasses. And then this is an example of what one of the high management plots look like. We have a lot of native clover coverage with some invasive grasses scattered in between there. And so what we were doing is we would stand over the plots and visually record percent cover and richness. So this is the data collected from those preliminary pre-burn surveys. So what we have here is a PCA plot, so a principal component analysis plot of all species abundance and richness values across blocks between management histories. So we have high management here in pink and low management here in blue. So axis PC1 indicates native versus non-native species with positive values associated with non-native species. And then we have PC2, which indicates life form, so forb versus grass, and positive values are associated with forbs. So what we have here is in the low management plots, we see that they're more associated with non-native grasses, whereas our high management plots are more associated with native forbs and grasses. So this essentially shows that our management plots are indeed different, and that our high management plots do have more native coverage compared to our low management plots. And then the same thing could be seen in these box plots. So these are box plots showing the distribution of native species richness in A, and then native species cover in B between the management histories across blocks. And again, we could see that overall high management has higher native species cover and richness compared to their low management counterparts. And so in October 11th through 12th, what we did is we went back to the field to collect litter. So we were collecting litter in order to estimate fuel loads. So first what we did is we took height data. So we recorded the 3 tallest plants in our 1 by 1 foot quadrats. Then what we would do is have it placed in the plot, and what we do is remove all the litter in there and put it into a paper bag. Then we would take soil core samples to record our pre-burn soil composition, and we repeated that process 4 times per plot for a total of 320 samples. And as a result, we had a Prius filled to the brim with samples. And so October 12th through 13th, I mean the reserve managers, went around and mowed all the plots. So they mowed buffers between 3 to 6 feet. And then we also tagged all the plots with yellow or pink tape in order to distinguish visually just across the field which ones were burn plots and which ones were control plots. Then what we did afterwards is we raked away the mowed litter to make sure that fire didn't spread across the reserve, because that would be really bad. And then the exciting day came where we did our prescribed burn. So October 18th through 19th, we did our burn. My advisor, Justin, pictured here, and one of the reserve managers was in charge of lighting. And then we also had people on holding with backpack sprayers, people managing the hoses, people recording data, and CAL FIRE was also there to monitor in case anything went wrong. And then what we also did was we recorded weather data before starting each fire. We recorded the duration of the fires, estimated flame length during the burn, and then the first burn itself was about 4 hours. So between 12 p.m. to 4 p.m. is when we burned. And then the second day was only about 2 hours since we had less plots to burn. So it was from 12:30 to 2:30. And so this is an example of what one of the high management plots looked like before the burn. And then this is that same plot after the burn. For the most part, all of our high management plots, except for 2, were mostly burned to ash. And all the low management plots were burned to char. And I posit the char is likely due to the density of grass. And those 2 high management plots that didn't fully combust had a lot of grass in there. So that's why I think they didn't really have the chance to burn to ash. And we have our low management plot. So this is what it looked like before the burn. And this is what it looked like after the burn. And then October 20th, I went back and I collected post-burn soil samples from all of the burn plots. And so then January 17th, we went back and collected soil for our seed bank study. And so we collected soil to analyze the immediate effects of prescribed fire on the transient soil seed bank, with continued vegetations assessing the persistent soil seed bank. And with that, we ended up collecting 9 soil samples per plot for 360 soil samples. And what we used here was just a bulb planter and just cored out soil cores from the ground from inside the plots. And then January 17th, again, that same day, is about 3 months after the burn, and you can see the difference between the burn and the control plot. So right here on the right, you can see it was burned, and over here on the left, you could see a lot of that standing dead litter. Again, the same thing here. This is one of the low management plots, and it's a lot more obvious to see this was the area that wasn't burned, and this was the area that was burned. And then the same thing here and as well here. This is just a further away photo, and you can very clearly see that this was burned, that's not burned, that's burned, or this is burned, and that's burned. And then I had the opportunity to go back out in the field earlier this week. So March 2nd, I went out to go see how things were germinating, since we got some early rains in a warm period and then more rains and more of a warm period. So just to make sure I don't need to start my field season earlier, but this is about 5 months after the burn. And again, we can see that difference between burned versus unburned plots. And so far, we overall see a reduction in non-native species in the burned plots. Like this plot here, it's a little hard to tell, but there's a lot of Senecio vulgaris here in the unburned control plot compared to the burned plot, where we see a lot less of it popping up. And then we also were able to see a lot of new species we either couldn't see in the plot because of all the standing dead litter, or because they were unable to germinate prior to the burn. And then we also see a lot of the plants that are starting to grow up are a lot bigger in size due to that lack of plant competition and dead grass litter standing in its way. So in terms of future plans, I plan to finally analyze the pre and post burn soil samples. We still need to weigh and calculate the fuel loads. And then this coming spring, so around April-ish, conduct our first round of spring vegetation surveys. So this will be the first round of post-burn surveys. And then in the fall, start conducting that seed bank study on our seed bank soil. And with that, I would like to thank everybody who has helped me so far in conducting the burns, because that was a lot to pull off. And I'm ready to take any questions.
Great job. Thank you for that talk.
Thank you, Keikoa. Does anybody have any questions? Katie, go ahead.
Hi, Keikoa. Thank you for this great talk. I have a question for you as well. I'm curious to know about the seed bank and the soil cores that you took. I realize you haven't done it yet, but I'm just curious to know what you expect to see. What kind of patterns do you expect to see?
I'm honestly not too sure, because part of the problem was we got some early rains before we burned, so there were some plants already germinating, and then more rains came during the winter and there was just time I couldn't go out there. So I'm sure there are plants that I might have missed just due to the whole timing of everything. So I'm not really sure what exactly we'll see during the seed bank study.
Anybody else have a question? There's a couple comments in the chat. Doug Stone says, what an amazing study. Thank you, Keikoa. And Radhika says, great presentation and location to study. Thank you so much, Keikoa. We really enjoyed your talk.
Thank you.
Yeah, thanks. And we would love to hear a follow-up if you ever have an opportunity after you've done more work. We would love to hear what happens. Thank you.
Of course. Thank you.
All right, we're moving on to our final speaker. We're going to hear from Belana Marahu, pardon me, who is our undergraduate scholarship recipient. She is a student at UC Davis studying plant sciences. Her research investigates the mysterious germination of rare and endangered vernal pool grasses in the tribe Orchidaceae. By exploring why chaff extract treatments enhance seed dormancy break, she hopes to unlock new strategies for propagating some of California's rarest plant species. Her work could play an important role in strengthening conservation efforts for threatened vernal pool habitats. Please welcome Belana.
Thank you for the introduction, Letitia. And hello, everyone. I'm Belana, and let me perhaps share my screen. There we go. And let's reopen a cold case together. This is breaking seed dormancy in rare vernal pool grasses. I'm sure many of you are already familiar with vernal pools. If you are, let me know which ones are your favorite. I would love to hear it. For anyone that is not, vernal pools are these seasonal wetlands that are completely rain dependent for their water source. And they're made up of these like alkaline clay soil beds that allow these pools to fill up during that wet season. And during that wet season, under the water, it is typically low nutrient and low oxygen. And in general, these are really harsh environments because of that one rain season. And then we end up with these absolutely stunning blooms of most of the time endemic species because of the specialization required to survive in such harsh conditions. And I'm focusing on species from the Sacramento and San Joaquin Valley, but this is a distribution of vernal pools all across California. And you might have heard about vernal pools recently because CNPS is suing Sacramento County for approving the development of vernal pools in Coyote Creek. Your chapter has also done a field trip to a vernal pool before, so some other ways you might be familiar. This lawsuit was rather recent. It was about 3 months ago that it was filed, so I don't think there's been any settlement, but the pools are in general extremely threatened because they are seasonal. So when they're not in that blooming season, they kind of just look like these flat beds of nutrient poor soil, and that can allow developers to get away with approving development even though they are full of all of this rich biodiversity. So with this extreme threat, we also then get a lot of endangered species. So my the species that I am working on are part of the Orcut tribe of grasses, or the Orcutii. This is a group of 9 species. I was part of the collection of 4 of them, or Tuctoria greenii, Orcutia niqualis, Orcutia tenuis, and Neostapia clucena. Like all grasses, their dispersal method is primarily from shattering. There are inflorescences from the rachis, and so you end up with these like individual florets to spikelets and a really limited dispersal range. And so where they occur in vernal pools, they are incredibly dominant. And that makes the mission of conserving vernal pools requiring conserving these species as well. This is, as I mentioned, these are both state and federally listed rare species. So this was collected under the California Plant Rescue Project, which I work for under the Center for Plant Diversity. And the standard protocol for this project is to scout, and then we collect seeds and voucher specimens and upload that data. And then we take those seeds back to the herbarium where we will clean and count them. The typical protocol requires that we clean the seeds till they are naked, which just means that we're limiting plant material that gets sent to seed repositories, because this ensures that we can do like more quality control, we can prevent pests like introduction. But in the case of the Orcutias, we want to, or in the Orcutii, we want to keep the chaff attached. So when we're counting them, we're trying to keep them inside of their Palea and Lemma little envelopes. And the reason why this has been customized for these groups is because of previous research where it has been found that like the chaff is really necessary to promote germination. And if we want to later restore this habitat, we need to be able to germinate these species. So in addition to finding out that chaff is really helpful for the germination of the Orcutii, we also know that they have a physiological dormancy, so they require a 4-week cold stratification. So this was back in like the 80s, and then this was revisited in the 2000s, where we now have new research that shows, yes, the chaff is still important, but sterilization has an additional improved germination rate. But all of these studies had different methods of going about off-site germination. So some did artificial vernal pools, some was filter paper and petri dishes, some did agar. And in general, we have some conflicting results and some kind of a lack of consistency with the protocol. So the trail goes cold in 2018. But now that leads to the research question of what is going on here? Why is the chaff important? So we ran a pilot study where we just wanted to see if the idea that chaff promotes germination still held up with all of the species that we had collected. So this was with Tuctoria greenii and Orcutia niqualis. We made what is called chaff extract, which is the plant material like the chaff that I described before, the Palea and Lemma, but this also includes like stems and leaves, just any plant material that we had. And that was soaked for 24 hours and shaken. And then we used that to inoculate some of our treatments. And this was to compare naked seed to ones with chaff attached. And we essentially found like, yes, this agrees that like chaff does promote germination. But that wasn't satisfying enough because this is something that we knew. So the research question that I will be focusing on today is if we can break the dormancy in these species with the same protocol. We hypothesized that yes, we would be able to do this because we're all in the same tribe. It makes sense that that would be the case. We know that chaff is important in those cases. We also hypothesized that chaff extract would be the most successful germination procedure if that were the case. And then our second question is, why is chaff extract different then? We suspect that there's a beneficial microbe involved. This was approached with a 96 well microtiter plate. So this is moving away from the agar plates. The purpose of the 96 well is that we could maximize our sample size and also eliminate variables because we could put one seed in each well, and then we could look at like an individual seed count with each treatment. This also eliminated variables from like soil because you're only looking at like the seed in its liquid environment. So the treatments that we decided to perform were sterile water as a control, GA, which is a growth hormone often used to promote germination, chaff extract, which I mentioned how we made before. And this is where we kind of diverge. We then took some of that chaff extract and pushed it through a 0.2 micron mesh filter, and that removes all of that's a physical removal of all of the microbes in that chaff extract. So this was all of the environmental chemistry that is in chaff extract minus the microbes. And then we also made acidic water to see if perhaps the chemistry, like the role of the chemistry, is specifically breaking down the seed coat or like dropping the pH in these alkaline environments. And so this was water that we made the same pH as the chaff extract. And then our last treatment, which was more on the side, it's not actually on the same plate as this other assay, but that was the growth hormone without cold stratification, because the goal of the growth hormone was to potentially create a positive control of like forcing germination outside of natural conditions. And there's reason to believe that GA could potentially remove the cold stratification. So I just wanted to see if that was possible to eliminate the cold stratification step because that would save 4 weeks in this protocol. So then this was all plated for each species with chaff extract made for each species. And you can see a clear difference in like just the physical appearance of each treatment. The chaff one is a little foggy and obviously brown. The filtered is not foggy, and that is like a good sign of like this one has microbial activity and then the other one does not. So the results, we had so we have a couple graphs here, but the x-axis is the days after removing from cold stratification and moving into the growth chamber, and the y-axis is the total seeds that were germinated. I will point out the scale on the y-axis is different because they had different germination successes for each species. But in Tinctoria greenii, we see that all treatments performed relatively the same, and then Orcutia niqualis, the chaff, was completely like was much more successful than our other treatments. And then Orcutia tenuis had more of a mixed result. So our takeaway of this is that our first hypothesis that we could use the same protocol to germinate all of the species was not supported because we actually couldn't germinate Neosdaptia calusina at all. And we think that this is likely due to a physical dormancy in the seed. It's also the least related to the rest of the tribe. So that kind of lines up with like the phylogenetic evidence that we have. And then Tinctoria greenii, of course, does not require the same laborious protocol as the Orcutias did. And then our second hypothesis that the chaff extract would be the most successful was somewhat supported because Tinctoria greenii, again, did not have a significant dependence on this microbial environment, but the Orcutia niqualis and Orcutia tenuis did seem to have a much stronger dependence on the microbes that are present in the chaff experiment that are not present in like the filtered experiment. So the overall conclusion is that Orcutia seeds should be stored with their chaff material. So this was a great justification for continuing with the protocol that we had for the seed counting. And that germination protocol and relationship with microbes from the environment is highly species dependent. And this is a great case study for why in conservation we should be looking at like species-based protocols. Yeah, and that the microbial factors promoting Orcutias are not the same as Tinctoria greenii. So thank you all, and thank you for the support from the Santa Clara Valley chapter of CMPS, and everyone else that helped make this possible, especially the people in Lab Laveau, which had been hosting me for the lab part of this research.
Wonderful. Thank you, Belana. Does anybody have any questions? Ooh, Doug.
Hi, Bilana.
Hi, Doug.
Thank you for a great talk. I have a question and then an observation. Really cool study. I'm just kind of curious if you experimented at all with using the chaff extract from one species, and if you cross if you cross if you crossed the experiment, like use the extract from one species to try to promote germination in a different species, was that successful?
That is a fantastic question and one that I did expect to get. I have not personally done it, but I think Carol actually might have something to say about that. Um, I have not done it, but it was something that I considered looking into. I potentially could with some of the Orchideas. I think it would be interesting to see if because we actually don't even know if there's like population-based differences. So I recently was making the chaff extract for two populations of Victoria greenii, and I was going to compare those, which I think is a good start to that.
Nice. And
So
then,
can I jump in and add something to that? This is Carol.
Yeah, go for it.
of course.
Doug, the Botanic Garden had some Orchidia pilosa that was naked, and they couldn't get it to germinate. But I, in the meantime, had sent them a whole bunch of Orchidia viscida to do some germination testing on and to put into permanent storage. And they used chaff extract from the viscida to get the pilosa to germinate.
All right. So that's the answer.
There you go.
Yeah.
Thank you, Carol.
Thanks, Carol. And then I had an observation. So Travis Columbus and I also did a germination experiment on some of these species in, I think it was in 2020, that by and large failed miserably.
Sorry.
But the amazing thing, especially in light of your results, is that the only plants we were able to grow to maturity, we grew 2 Neostaphias from seed to maturity in the greenhouse, and it was kind of an uncontrolled experiment. So we don't actually know what we did to get those 2 plants to grow like that, but they did.
Well, if you learned anything from that, please let me know. Because right now we're planning to like dump some boiling water over Neostaphia and like file it down. So I'm open to hearing what methods you tried.
There's someone named Heather Davis who used to
Yes.
work for,
Continue. Yeah.
I'm trying to think of who she used to work for. It was somewhere out of Sonoma State. She was a tech for them. She managed to get Neostaphia to germinate because she did some pollination distance stuff out at Jepson Prairie, where she actually had the Neostaphia plants that she had grown in little diapers, so that they, had their own moisture source and stuff like that. So there's got to be something out there about
Yeah.
the Neostaphia germination.
I tried to find her protocol for it, and I believe it was like an artificial vernal pool kind of. It was like a much more intensive method.
Yeah, I think she used a mesocosm, basically a kiddie pool. Um, but I remember her sending around a whole bunch of pictures of the fungus emerging on the chaff when she finally got them to germinate. So there was definitely some fungal activity going on.
I did perform the same assay that I showed on the Neostaphia clucinate. There wasn't any germination from it, but yeah, there's a lot of like fungus that goes on there, some interesting yeasts that come up. Um, and that was like a similar situation that Griggs had where he like saw this fungus grow on there, and so that was like why he thought that there was like a fungus related to the germination. But if I if I do get it to germinate, I will be culturing, and that's something that I'm working on right now, is culturing some of the chaff extract to see if we can figure out what the microbe is.
Have you seen that paper by, I think is Jorge Montiel, about microbial diversity in vernal pools? He was using, I think, an environmental DNA approach to gain some insight into that. But you know, at some point, we might want to try to connect up these different
Yeah.
approaches.
Yeah, I mean, so if I can tease what I'm working on right now, I used the paper that he had written oh, can I go to the next slide? So I'm doing some selective media to see. So I looked at his list of like microbiota that he found, and like also was looking at which microbiota are known for already helping with germination. And so these were the genera that like lined up between those two lists. And so I'll be selecting from that because these ones are ones that we know do occur in vernal pools. So we'll see what that yields, but that is something that I'm hoping to do.
Keep us posted, please.
Yeah.
Thank you.
Like Justin has a question.
Yeah, thanks. That was such an interesting talk, really excellent work. I'm just wondering, so like if you think it is microbial in nature, like have you thought about maybe using soil extracts as well, like in some of your assays?
I imagine some of the soil kind of gets in there because the thing with the orcuts is they're sticky. And so like soil kind of naturally traps onto them. I have not included it into the assay. That would be interesting to do. I'm kind of sticking it with the chaff extract for now. But I think I was told like if I can get a soil sample, I would be open to it. But I haven't done the RAMS application yet.
Yeah, no, that makes a lot of sense. I'm just, you know, it's really important to do the work that you're doing like in artificial settings so we know like what the germination cues are, but we also have to think about like what is happening in the field. And I'm just curious like what role the soil microbiome might play in that. But yeah, really excellent work.
Thank you. And thank you for bringing that up. It is a good point. The soil is like a messy spot to be in though.
It's very messy, so you might want to stay focused.
Well, thank you, everyone.
All right. Thank you, Valana. And I actually have a few closing slides for the evening. So I'm going to go ahead and share them. Excuse me, I can find them in just a sec. Okay, let's see. What am I doing here? So I just wanted to thank everybody for joining us, and especially a big hand for all of our students and their amazing projects. That was wonderful. Really appreciate you guys putting in the work to put these presentations together. If anybody wants to learn more about our scholarship program, you can go to our website, and there's a section there about the scholarships. We do this every year, and we can use your help to do it. So if you would like to make a donation to our scholarship program, you can go to our website or go to that QR code, but cnps-scv.org/support-scholarships. If you would like to help us make more of these things possible, we would very much appreciate it. And then finally, oops, where did it go? I don't know. There. I just wanted to remind everybody that there is a recording of this talk. It's available right now on our YouTube channel. So if you want to go back and look at something, or if you want to share it with a friend, please feel free to do that. It's available right now. And I will stop my share. And I just wanted to say thank you to everybody for joining us. And I guess that concludes our evening tonight. So thanks again, everybody. And I'm going to end the Zoom now. Good night.